So I have chapter 7 and 8 kind of combined together. So what we want to talk about here is microbial genetics. So how bacteria replicate their DNA, how they mutate, um, how they're used in biotech a little bit at the end. Okay, so the first thing is what are chromosomes? And I think you guys know what a chromosome is. So chromosome is typically double-stranded DNA. It's a way that we organize our DNA. It's the way that we have our genes organized, I guess. Okay, now prokaryotic chromosomes. So in prokaryotes, so in bacteria, most only have one chromosome. So it's important to remember that they only have a single copy of each gene. So humans and other animals are diploid, right? So we have two copies of each gene. We have one from our mom, one from the dad, right? So we end up with two copies, so that's called diploid. But bacteria are haploid, so they only have one copy. So their diploid is also referred to as 2N, so 2 times the number of chromosomes that you have. Haploid is 1N, so they only have one copy of everything. Okay, it's important to remember, and we've already talked about this before, that bacteria also may contain plasmids, and we'll talk about plasmids more in a minute. And the nucleoid is the region where the bacterial chromosome is found. So here, I think this is E. coli, and they did a TEM, and they colorized it. So this is the chromosome that's dividing through binary fission. The cells are dividing through binary fission. Okay, so prokaryote versus eukaryote. So I have a picture over here. So prokaryotic DNA is circular. Eukaryotic DNA chromosomes are linear. Okay, so that's the big difference. How they replicate, everything replicates at this thing we call it an ORI. So the ORI is referred to up here as it's the origin of replication. So you guys remember replication is just when we copy DNA. So when we go from DNA to DNA. Okay, so replication occurs at this origin. So origin of replication, ORI is origin. Okay, origin of replication. So I have a little chart here. Location of the chromosome. So in prokaryotes, it's in the nucleoid, and eukaryotes, it's going to be in the nucleus because they have membrane-bound organelles. Shape. Prokaryotes, it's normally circular. There are exceptions, of course, right? And then eukaryotes, they're linear, right? We always picture them as this X. And number of chromosomes, typically prokaryotes are haploid, or they only have one chromosome. Of course, there are exceptions, so some things have two, some have a linear chromosome, which is weird. Okay, the number of chromosomes in a eukaryote is variable, so it varies depending on the species. Sometimes they're diploid, sometimes plants are not diploid, but typically things are diploid and they have two copies of a gene. Okay, so microbial genomes, so I want you to remember that genomes, let's see if we can, genomes, it's the total DNA content of a cell. So it's all of the DNA that's in the cell, that's the genome. So they've sequenced the human genome, they've sequenced lots and lots of genomes, and you can get online and you can find them. There's a website, it's I don't know why I have yellow, but NCBI. So it's like the National Center for Biological something. Okay, but NCBI has lots of genetic information, lots of databases. So, let's see, we'll go down here. Okay, so they have databases of the genomes and like all sorts of information. So the human mouse genome are 3 billion base pairs. Okay, so remember base pairs are like A paired with T or C paired with G. So when we're talking about DNA, the units that we use are base pairs. Okay, so they're talking about megabases. So a megabase is 1,000,000 bases. So in microbes, so like bacteria, typically the genome is less than 10 million base pairs. So they've done a lot of work on sequencing them. Okay, so trying to figure out what A's, C's, T's, and G's are there and what order they're in, so the sequence. So E. coli has been studied extensively. It's the model organism for bacteria. So they say that it has 4.64 megabases, okay, and that within that there are 4,300 genes. Haemophilus influenza is another organism. It's a lot smaller, so it's 1.8 megabases. So when they sequence things, and this is a sequence, I think this might be E. coli, but they'll make a map of the chromosome. So with humans, right, the map is linear. So with bacteria, the map is going to be circular. And each of these little things on here represents a gene. So each one of these abbreviations is where a gene would be. In this little region right here, there's a crazy amount of genes found. So they take that little piece and then they blow it up so you can see what's actually there. So it's kind of cool. So somebody knows what each one of these genes do, or they think they know. Okay. So how does bacterial replication occur? So how do they replicate their DNA? So bacterial replication is semi-conservative, just like human chromosomes are, right? So you keep the old strain and make a copy. Origin of replication is where it begins. Okay, and then there's a whole list of enzymes involved. Okay, so because it's circular and it's double-stranded, it's tricky. The winding and the unwinding is a little tricky. So they have helicase, and helicase is going to unwind the double helix so that we can copy the DNA. Primase is an enzyme that's going to form RNA primers to get the DNA replication started. RNA polymerase 3 extends the DNA so it builds it. Polymerase 1 removes the primers because we don't need them anymore. Ligase is going to glue our pieces of DNA together, so it kind of seals it up. I always just think of it as a glue stick. Okay, gyrase, so gyrate, it's going to recoil the DNA. So it actually kind of nicks it and twists it. And then another enzyme is topoisomerase. And topoisomerase, if the DNA coil isn't wound properly, topoisomerase can come in and fix it. Okay, now these things, okay, especially gyrase, topoisomerase, these are targets for antibiotics. Okay, so if an antibiotic can go in and it can interfere with gyrase, it could interfere with this topoisomerase, it can stop the bacteria from replicating, so it can control a pathogen. Okay, I want you to be familiar with these, like you should know polymerase builds DNA, ligase is the glue, gyrase recoils. I want you to know basics, but you don't have to, I'm not going to make you write an essay or anything on it. Okay, now, why is the DNA coiled? Okay, so the supercoiling of the DNA occurs so that the DNA will actually all fit into the little cell. So someone did an experiment where they lysed a bacterial cell using osmosis, so it's kind of gentle. And when they did, they let all of the DNA out and it unwound. And it turned out that the DNA was 800 times longer than the little cell actually was. So that's how much DNA has to fit into those tiny bacterial cells. So the DNA has to be supercoiled in order to fit in there. Now, when we make proteins, and we're going from DNA to mRNA a protein, right? We're doing transcription and translation. We have to also unwind it in order to read it to make proteins. So the supercoiling actually will regulate what genes are being expressed or what proteins are being made. Okay, now other genetic elements that are found, the big one that I have on this slide is plasmids. So here's a picture with the bacterial chromosome. Okay, so circular chromosome. It's got all the genome in there. And then the plasmid. Okay, so plasmid is extra. Okay, so the plasmid is double-stranded. Okay, it's circular, but it can give important properties to the bacteria. Okay, so it can code for the production of different proteins. Okay, so its DNA can be read, and it makes proteins that give the bacteria with the plasmid an evolutionary advantage over others. Okay, plasmids aren't viruses. Okay, so they're not viruses. They don't typically cause damage to the cell. They don't exist outside of the cell naturally. Okay, we can make them and we have them, but it's not like a virus that can hang out and cause an infection. Okay, so they're not viruses. Okay, they're not chromosomes because they're not essential for the cell to function. Okay, a plasmid can be removed. Okay, plasmid can be kicked out, and the bacterium will survive without it. Okay. Okay, now, one important type of plasmid that is medically important are resistance plasmids. So resistance plasmids carry resistance genes for antibiotics and other drugs. So here's an example of a resistance plasmid, so a R plasmid. So sometimes people call them R plasmids, sometimes they call them R factors. So every place where they have a color like this is where something's happening, okay? So here's where, this is the origin of replication. Okay, so that's the ORI. Then we'll talk about this resistance transfer factor here in a second. Okay, but this green spot and this green spot contain a gene, right, that makes a protein that gives the bacteria resistance to mercury, lead, arsenic, and antimony. So those are really bad compounds that normally would kill bacteria. Okay, but if they have this plasmid with these genes in them, these chemicals can't kill them. Okay, this plasmid also has resistance to some antibiotics. Okay, so it has three genes in it. So one is resistance to erythromycin, one is resistant to penicillin, and the other one is to chloramphenicol. So I want you to get the idea that these are genes that are coding for proteins. Okay, and the proteins are typically an enzyme, and that enzyme is going to break down penicillin. Okay, so that's how that works. Okay, so I have two components, resistance transfer factor. So resistance transfer factor is going to, it's a gene that is going to allow this plasmid to be transferred to other organisms. Okay, so they can be transferred between species. Okay, so you could have E. coli give a resistance gene to Staphylococcus. Okay, then here's our genes, and this is what I just tried to explain to you. So our genes confers resistance to specific antibiotics or disinfectants, and it directs synthesis of an enzyme. Okay, so resistance plasmids can have up to 10 genes in it. They can be transferred from organism to organism. So here's the pilus. Remember pilus from before? Okay, so this can be transferred from one organism to another. So the transfer is rapid, and it can occur within a species or between genera. Here is a video of a really cool experiment. It was done in 2016 at Harvard, where I want you to watch it. They made a mega plate, and they put different antibiotics in the plate, and then they inoculated the plate with E. coli, and they watched as the E. coli moved across the plate through more concentrated antibiotics. So it's kind of crazy to watch, but it's interesting. It's cool. It just shows you how fast a mutation and the evolution of bacteria can occur. So resistance to antibiotics can occur really fast if they're exposed to antibiotics. Okay, now another genetic component. So I want you to think about, okay, so in eukaryotes, we have lots of genetic diversity because of the way we reproduce, right? I can't spell diversity, but you know what I'm trying to say. Okay, so we have lots of genetic diversity because of the way we actually reproduce, right? Because we have sperm and eggs, and then we pick our mate, and then there's recombination. Okay, now bacteria, okay, so prokaryotes, right? Their reproduction is asexual. So they don't have the opportunity to have a lot of recombination of genetic material. And the recombination of changing of genetic material is important for organisms to survive. Okay, so one way that bacteria can move information around are these things called transposable elements. Okay, so they also call them transposons, they call them mobile genetic elements, and they used to call them jumping genes. Okay, so they were discovered by Barbara McClintock, who was, I always mention her because she was a female scientist before there were a lot of women in science, and she was at Cornell. Okay, so she noticed, so what she actually worked with was corn, and the Indian corn that's the different colors, like all the kernels are different colors, that's what she worked with. And she discovered that the colors were not being inherited the way Mendelian inheritance would have predicted. So something weird was going on. So she found that some organisms have genetic information, they have transposons, so there's genes, and they have a system where things can actually be moved around, okay? So genes can be transferred from one organism to another. They could go from a plasmid, they could go from a chromosome to another chromosome. So they can hop within a chromosome, maybe not within organisms, okay, so I shouldn't have said that. Okay, they can hop within a chromosome, and they can hop from a plasmid to a chromosome. Okay, so they're staying within the chromosome. They're not going to somebody else. Okay, now, transposons, why we care about them, is they can carry antibiotic resistance genes. Okay, so I have a link to a transposon video which shows how that information actually moves around. So watch that. You don't have to memorize all the steps, but I want you to get the gist of it, the overall concept of how it works. Okay, now what's a mutation? So a mutation is a permanent inheritable change in genetic information. Okay, so it's a change in genetic information that can be passed on. So what it's doing is it's altering the A's, T's, C's, and G's, right, the base sequence of the DNA. So there are three main ways that can happen. Okay, so if I make A's, C's, T's, and G's, okay, one thing that could happen is we could have a base substitution. Okay, so we could have something put in instead of A, T, C, or G. Okay, we could have an insertion or a deletion. Okay, so we could have something stuck in there that's not supposed to be there, or we could have something removed accidentally. Okay, and then the third thing is we could have an inversion. So instead of having A, T, C, G, we get an inversion. So we get A, C, T, G. Okay, now, why do we care? What's going to matter? And you guys know about transcription and translation. So why it's important is because these sequences of 3 nucleotides are going to code, right, for amino acids. So if the sequence is messed up, we may not get the right amino acid and we may not get the right protein. Okay, whoops, this looks funny. Okay, so what happens to proteins when a mutation occurs? Okay, so sometimes the code will change. Okay, so say we have A, T, C. Okay, sometimes we change the C. Maybe this got changed to another T. The code might change, but the amino acid doesn't. Okay, so you have to remember that we have 64 possible codons, but we only have 20 possible amino acids. So there's more than one way to get an amino acid. Okay, so we can have change that occurs and it causes a silent mutation. So the code can change, but the amino acid doesn't. Okay, the second thing that could happen is a nonsense mutation, or a missense, sorry. So missense would change the protein, so it changes the amino acid that's here. So maybe instead of this being, if this is leucine, okay, and this is leucine, but if we make it a G, maybe we make it, I don't know, isoleucine. Okay, so we can change it. Okay, there can be nonsense mutations. So a nonsense mutation would mess up would create a stop when we don't normally have a stop. And then you can have frame shifts. So a frame shift would be if this is what well, I should have made this something else. Okay, let's make this a T and a C and a G. If this is normally what we have, right, and this disappears, and okay, so this is normally what we have. So we'd have ATG and we'd have CTC. Okay, but if we get rid of this, right, it's going to cause us to move this over here. And that's going to code for a different amino acid. Okay, so it's all about what proteins we're making. Okay, so I have this picture. So here's what we're looking for. So AAA and UUU, and that codes for phenylalanine. Okay, if we have a substitution here, that could give us leucine. Okay, but if we put a G back in, if we have a different substitution, okay, so this would be missense. Okay, but this gives us the same as this, so this would be silent. Okay, and this is supposed to show you what I just kind of tried to draw for you. Okay, so there's normal. If we have a deletion, we lose something, right? And if we have an insertion, we put something in. Okay, now what can cause a mutation? So a mutation could be spontaneous, so it's just a mistake in replication that our polymerases don't catch. Okay, but there can also be induced mutations. So induced mutations occur if we're exposed to a mutagen. So a mutagen could be a chemical or it could be a physical agent that's going to damage your DNA. So some examples of chemicals would be things that are base analogs. So they might look like a base. So they might look like a nucleotide. And caffeine is actually an example of that. Chelating agents. So an example is this nitrosoguanidine, which is actually pretty nasty. And it'll add a methyl to the nitrogen base. So then it can't be red. Deaminating, remove amino groups. So then they can't be red. And then something like atabrine. So atabrine can actually, I have a picture of it up here, actually gets inserted into the DNA, and then it can't be copied. It sometimes it used to be used to treat malaria, but because it has side effects, they don't. It can also be used to treat giardia if someone can't take Flagyl. Okay, so those are chemical. Physical agents, examples would be radiation, so UV light. UV light actually, when it damages, it links pyrimidines together, so it makes these things called pyrimidine dimers. X-rays and gamma rays form free radicals, and so they just kind of bounce around and they damage DNA, they break it apart. Okay, now, genetic exchange in prokaryotes. So typically, right, if we have binary fission, okay, the daughter cells are exactly identical to the parent cell. Okay, so there's parent, there's the daughter. Okay, and they're the same. They're genetically similar. They're going to be exactly the same. They should be copies. Okay, so this is vertical gene transfer. Okay, now bacteria have horizontal or lateral transfer. So they have transfer within a generation. So it'd be like you passing DNA onto your sibling, okay, without having sex. Okay, so it's not inheritance, it's not reproduction, but they can swap DNA. They can move DNA around. That gives them an advantage evolution-wise because they can change, they can be modified. Okay, so here's 3 mechanisms of lateral movement. Okay, so the first is transformation, the second is transduction, and the third is conjugation. And I have slides for each one of these. Okay, and actually, I think I'm going to stop here and upload it because, excuse me, because I think that thing is getting long.